Okay, so the muscular system. We are going to talk a lot about muscle tissue. Remember the labeling of the muscles, which is also something that will not be on the next exam, is an optional quiz that you can take. I've mentioned it any number of times. If you need more information on it, come see me, okay? That's not in this next exam. So, you probably knew coming into this class that muscles contract and allow you to move your body. So muscles are responsible for all types of body movement. It isn't just the skeletal muscles that contract and move our bones. We also have cardiac muscle, which we find in our heart, and we also have smooth muscles that we find in our hollow organs. For instance, our urinary bladder. The whole length of our digestive tract has smooth muscle in it. And there are isolated other areas like your blood vessels and the ureters in the female the urethra. No. The fallopian tubes in the female reproductive system also have some smooth muscle. We need to understand that there are three basic types of skeletal muscle, types of muscle: skeletal, cardiac, and smooth. Now, for the tissues chapter, I wanted you simply to know that list. Now we're going to expand on your knowledge of those three types of muscle tissue. Before we get into the specifics of the three types of muscle tissue, let's talk about the characteristics of muscle. Skeletal and smooth muscle cells are elongated. They're very, very long. As a matter of fact, when you talk about the quadriceps muscle that goes down the back of the leg, they are, actually, I think I meant to say hamstrings. The hamstring muscle that goes down the back of your leg. They attach on bones in the pelvic girdle and also attach on leg bones. That muscle cell is very, very long, isn't it? That is a skeletal muscle cell. It's incredibly long. Smooth muscle cells are also somewhat elongated, but not usually quite as long as the skeletal muscles. And what we need to understand is now we're going to call muscle cells muscle fibers. Same thing. A muscle cell is what we call muscle fibers. Muscles can contract, and when they do, they shorten. And here's how that works. My biceps muscle, that's probably the most common muscle. Everybody kind of knows that this is your biceps muscle. It's attached up here in the shoulder girdle. It crosses, hair all over me, crosses the elbow joint, okay, attaches down here on the radius. When the biceps shortens and contracts, this is the origin of that muscle. This is the bone that's not moving. The insertion of that muscle crosses this joint, and when the muscle shortens, it puts pressure on the tendon that attaches down here in the radius and pulls the forearm up. So, contraction and shortening of the muscles is due to the movement of microfilaments. That's what we're going to learn about in this chapter. You probably knew coming into this class that your biceps muscle sits right here on your upper arm, and then when it shortens, you can pull up your forearm. You just don't exactly know what goes on inside that muscle cell. That's what we're going to talk about now. And then to do that, we need to talk about the microfilaments. And we will get to that. The story will unfold as we do this chapter. All muscles share some terminology. Prefixes of myo and mys refer to muscle. And then the prefix sarco means flesh. Okay. When we eat the flesh of a cow, we are eating cow muscle, okay? That may actually make some of you realize that maybe you want to be vegetarians now, okay? No, me either. Yeah, no. So, our muscles are our flesh, and very often some of the terminology associated in this chapter will start with sarco. Some of it myo, some of it mys. All of it basically refers to muscle tissue. So here is what the entire biceps muscle would look like, and we've made a cross section through it. The end of this muscle is going to form a tendon. It's actually the covering. There are 3 different connective tissue coverings in muscles. the epimysium is going to continue. The muscle cells end right here. The epimysium continues and forms the tendon that's going to attach to the bone. If we make a transection through this muscle, what we're going to learn is each individual of these pink circles here, we have gone through a muscle cell or a muscle fiber. And the muscle cells are bundled in groups called fascicles, and that's not labeled for yet. But one of the things we do need to know is that there are 3 connective tissue coverings covering the entire muscle group. That connective tissue covering is called the epimysium. Bundles of muscle fibers are held together in groups called fascicles. There is a connective tissue covering on top of that. That is actually called the perimysium. The covering around the fascicle is the perimysium. And then each individual muscle cell, this is one muscle cell, we're looking at it in a different direction now. That connective tissue covering on each individual cell is called an endomysium. If you have had medical terminology, you know that epi means around, so it's saying around the entire muscle group is the epimysium. Endo means within, so the furthest you can go in the structure of a whole muscle is to get to the muscle cell. We're going to go inside there too, but we don't have any connective tissue covered in there. So the endomysium covers individual muscle cells. And then peri kind of means in the middle. Okay, so in the middle of the epimysium and endomysium, you have a perimysium that covers the groups of muscle fibers into fascicles. And you're going to see the word fascicle as we go forward. Okay, so we're going to talk about gross muscle anatomy, and we're also going to talk about microscopic muscle anatomy. We're going to talk about what goes on inside, what we've got inside each muscle fiber in order to explain how contraction takes place. That's microscopic muscle anatomy, and then what we can see with the naked eye is called gross anatomy. All right, any questions so far? Now, here is where the book organizes for you information about the 3 different types of muscles. And on this particular table, and I believe the table goes down 2 pages, but most of the information that I want, oops, I don't want to do that. Most of the information that I want, do you not have that table? All right, does anybody need paper to make this page 182? You have, I do believe, most of the information that you need. Yeah, you have your pictures in it. So you've got this table here. But it may behoove you to recreate this table on a separate piece of paper. Would anybody like a piece of paper to do that with? Because I can go to my office and get some paper. Okay, I'm going to go get some unlined paper. I'll be right back. Okay, anybody here? All right. You good? Okay. All right. And Courtney, you were here for the quiz, right? Crystal. Oh, Courtney. You were, oh, afterwards, that's right. I'm getting you confused. Courtney, Crystal, Courtney, Crystal. Anybody else over here need a piece of paper? Everybody good? Okay, so here's what this table is telling us. There are 3 different types of muscle tissue. So across the top here you see skeletal, cardiac, and smooth. On the left-hand side, you see the topics that we're going to differentiate. Okay. So first we're going to talk about body location, then we're going to talk about the cell shape and appearance of the cell, and then we're going to talk about the tissue, connective tissue components. Okay. So for body location, the skeletal muscles are attached to bone or skin. For some facial muscles, that's why we include skin there. The only place we find cardiac muscle is the walls of the heart, and then the smooth muscles are found mostly in walls of visceral organs other than the heart. If you want to write yourself some examples, put the lumen of the digestive tract, or just plain digestive tract, urinary bladder, blood vessel walls. Those are all hollow organs, and we find smooth muscle there.
Can you say those examples one more time?
Sure. Digestive tract, urinary bladder, and walls of blood vessels. So, how are we doing with that category? The next characteristic is cell shape and appearance. Skeletal cells are single, very long, cylindrical. They are multinucleate. I need you to star that. Okay, I think it's very important to know that because a cell, when it's very, very long, it needs more than one control center in order to organize the metabolism that goes on inside the cell. So skeletal cells are multinucleate, and the other thing to star here are striations. It is striated. If you were to look at it under the microscope, you would see obvious bands. They're called light and dark bands, and they alternate. And we're going to see some pictures of that in a minute. So that's skeletal cell shape and appearance. Cardiac cells are branching, and when we look at the picture of cardiac cells, I'll point that out. Cardiac cells only have one nucleus in that cell, and it is striated. Also, it has intercalated discs. Okay, actually, everything in this this block is pretty important, but make sure you know that the cardiac cells are uninucleate and that they are striated. Then the smooth muscle cells, they are single cells. They are fusiform, which means that they narrow at each end. So they're bulgy in the middle, and then they narrow down towards the end. We'll see a picture of that in a minute. There are no striations. This must be Monday morning. My mouth is not engaged yet. Connective tissue components. Skeletal muscle has all three. Cardiac muscle only has an endomysium, and so doesn't smooth muscle. All right, I'll let you finish writing that particular part of the table for a second, in case you're still. All right, how's everybody doing? Still writing? Okay. Cold. I'm feeling a draft. I assume it's coming from that thing. You must be right underneath. Yeah, it's must be the air conditioning, which is kind of weird. Okay, how are we doing with? Still writing? Okay.
Which chart is it going to be on the test, or is the information from this chart is going to be on the test?
Both.
Okay, then if the chart's going to be on the test, is it going to be a blank chart, or is it going to be fill in the squares chart?
I...
Oh. Well, no, I just, when it's filling the square, sometimes I study by triggers, you know, like,
Yeah.
and where, if I have to just memorize it.
There's a lot of memorizing going on here, some learning, but I may have the table, and I may give you some of the answers in the table and expect you to fill in the others. Does that help you?
Yep.
Okay. All right, are we ready to go to the next slide? Let's go to the next slide. C. Now, I am only going to have you pay attention to the top characteristic, which is regulation of contraction. Speed of contraction and rhythmic of contraction, I don't need you to know that. So the bottom two characteristics, don't bother copying those. But this top characteristic, where it says that the control or the regulation on contraction is voluntary for skeletal, it means that we get to tell our skeletal muscles to contract. We call that voluntary control. Cardiac muscle, do we get to tell our heart to contract? No. It has autorhythmicity, which we will talk when we get to the cardiovascular system. Smooth muscles, do you tell your digestive system muscles, you got food in your stomach, start contracting? No, it's involuntary. Now, if you go back to between this row and the previous row that talked about, let me just go back, talked about striations, which is actually cell shape and appearance, I'm actually going to switch to let's see if there's any place I can write.
The chart in the book also tells us what controls the regulations, like via the nervous system and stuff like that. Do we need to know that, or just what's on your chart?
Just what's on the chart. Okay, I'm actually just going to write on this to show you how we can distinguish one type of muscle from another, between using whether it's striated or not and the control on the type of innervation. So I want to go to pointer options, and I want it to be a felt pen. And what I want to do is up here, voluntary striated. Skeletal muscle is voluntary control, and its appearance is striated. The cardiac muscle is an involuntary control, and it is striated. So see how we can distinguish right now skeletal from cardiac? They're both striated, but skeletal muscle is innervated voluntarily. We get to tell the muscle to contract. The cardiac muscle is involuntary. Now, smooth muscle, the control of contractions is involuntary, but so that would be the same as cardiac, but there's no striations. And that's why it gets its name of being smooth, because if you look at these muscles, because it doesn't have any striations, it looks smooth. Okay? So you might want to add that to that part of your table, just so that you have those together, so that you know how you can distinguish the three types of muscle tissue based on the type of regulation of contraction, or what I call control, and whether it's striated or not. Okay, and as Farah was questioning, I will expect you to know the information in this table. It will figure very importantly in terms of points on this chapter. Okay? Hint, hint. Make sure you know this table. All right, any questions on the information in this table? What I want you to know, what I don't want you to know. So your outline organizes this information again for you in a slightly different manner. Okay? It's telling us that skeletal muscles are mostly attached by tendons to bones. They are multinucleated, striated, which means they have visible banding, and voluntary, subject to conscious control. The connective tissue wrappings of skeletal muscle, we have gone over those already. So we know that there's an endomysium, perimysium, epimysium, and something called fascia, which I'll point out in a minute. So we see this diagram again, slightly different orientation. And it's labeling for you the group of muscle cells called the fascicle. So this entire piece right here is the whole muscle. Let's say it's the biceps. It's covered by a connective tissue covering called an epimysium. The muscle cells are organized into fascicles, groups of muscle fibers. There's a connective tissue covering wrapped around those. That's called the perimysium. And then each individual muscle fiber, see, that's coming out of the diagram. They're taking out one particular muscle fiber. Let me just change back to an arrow. Pointer, arrow. Right here, covered by an endomysium. The way your book labels it is to label right in between two muscle cells right here. Okay? And then we can also call this epimysium, as it attaches to structures around it, that's what we call fascia. Okay, connective tissue that connects two different structures together, muscle to muscle or muscle to skin. That's going to be the layer called the fascia.
So is the tendon made out of epimysium?
Correct. It is. It's made out of epimysium. Okay. And so I think I explained this in the couple diagrams ago. The muscle cells end here, but the epimysium does not. The epimysium continues and comes out of the muscle, forms the tendon, which attaches very integrally with the periosteum on top of the bone. Tendon connections are tough. Sometimes you will rip a muscle before you rip a tendon. They're that strong. All right. Any other additional questions?
So each muscular cell, the way they get components coming out, the white on the outside is the fascia, right?
Yes. Well, it is the, this is the epimysium forming the tendon. But see, fascia isn't labeled here. That's a fascicle. Okay, is that what you meant? Okay, what's coming out is an entire fascicle and then one individual muscle cell. Okay?
They're all individual fascicles, though, right?
Yes.
They just pulled out one.
They pulled out one muscle fiber. So this round piece right here, this whole group is a fascicle. There's many muscle fibers within there. Okay? This is one muscle cell or one muscle fiber. Okay? Everybody with me?
Oh, okay, fascicle. Okay, it was a different. I'm like, okay, wait a minute. I thought you said. So it's the same thing.
Okay.
I wrote it down.
All right, good. Skeletal muscle attachments. The epimysium blends into a connective tissue attachment. That's what they mean there is at the muscle cells end, epimysium continues and then blends with the periosteum. We have two kind of arrangements of tendons. The typical one that we're used to, like we saw in the picture previously, that's a cord-like structure. There's lots of collagen in there and often crosses a joint due to toughness and small size. We also have attachments of muscles in sheets, okay? And to show you what an aponeurosis looks like, Victoria, can I borrow your book for a minute? If we go look at, later in the chapter, if we go look at the diagrams where you learn where the muscles are attached, or the names of the muscles. So for instance, on 208, figure 6.18, you see a sheet-like tendon that's attaching the latissimus dorsi down the midline. Also in the anterior part of the body where you have the rectus abdominis muscles. So another good example is on page, on page 216. You see the rectus abdominal muscle, kind of one side's transected away, but on the other side where you see a lot of white, flat material, that is an aponeurosis that's attaching the abdominal muscles to one another. Sometimes tendons attach muscles to bone, sometimes they attach muscle to muscle. Okay, now I gotta bring you
So
back.
it's the white?
The white, exactly. Tendons are white.
Oh, so it is a tendon.
It is a tendon, but we typically use the word tendon when we're talking about the cord-like structure that would, for instance, attach my biceps across my elbow here. It's kind of roundish in diameter if you were to cut through it. That's technically a tendon. An aponeurosis is a flat form of a tendon, if you want to call it that way, but it's basically a muscle attachment
Okay.
mechanism.
How many tendons are there? Are there
Oh,
any different types of tendons?
No, not really.
Okay.
No. Nope. They're basically made of the same structure. We're just distinguishing between cord tendons and sheet-like tendons. Okay. So the sheet-like ones, we call it an aponeurosis.
So we must have a lot of that if we have 206 bones, right?
Yes. If you have 206 bones, you've got lots of tendons and lots of aponeuroses. Skeletal muscle attachments. Sites of muscle attachments can be on bones, cartilages, and connective tissues. When muscle attaches to muscle, another muscle, it's going to do so through the connective tissue coverings, okay, the epimysium. All right, smooth muscle characteristics. So we've finished with skeletal muscle characteristics. Now let's talk about smooth muscle characteristics. We learned from the table that they lack striations, they have spindle-shaped cells, and a single nucleus. Involuntary, no conscious control, found mainly in the walls of hollow organs. And so up here in the upper right, that's what a spindle-shaped cell looks like. So see how it is bulgy in the middle right here, but it narrows on both sides? That's what's meant by spindle-shaped. One nucleus, and we find them in the walls of our digestive tract. They are going to be in right here, okay? And they're actually in two different directions. There's some that are in the plane of this board, and then there's some that go perpendicular to the plane of the board as we look at them. We'll talk more about that when we get to the digestive tract. So that is what smooth muscles look like. Cardiac muscles have striations, a single nucleus, branching cells joined to another muscle cell at an intercalated disc, under involuntary control, found only in the walls of the heart. So this is showing us bands of cardiac cells in the upper part of the heart and in the lower part of the heart. We'll spend more time talking about that when we get to the cardiovascular system. But you know what? I don't have it. Let me see how quickly I can go get what I want, because it's not in my PowerPoint. I want to discard them. Yeah, let's discard them. All right, I can go to some other files that I have. Back here. I want to go to JPEG images. I want labeled. And I want what you have in your book. Tables. I bet it's in tables down here. Here we go. This is what I want.
I can bring that stuff up on my computer too, right?
Yep. Yes, you can. Okay, so I wanted to bring this up because you have this in your book, and it's the best way that I can explain how the three different types of muscle tissue are going to look under the microscope. Now, I'm not going to take you to the lab and show you it, so you're just going to have to assume that if you were to take out a microscope and prepared slides of these three types of tissues, this is what skeletal muscle would look like. And what you're seeing here is this one area in between, this a little bit of white space here. Everything between here is one muscle cell or one muscle fiber, and it continues on past this field. Remember, they're very long. Depending on how long that muscle is, that whole muscle group, that cell's going to be that long, which is why there's more than one nucleus, okay? And you've got striations. See how there's dark light, dark light, dark light? That's what's meant by striations, okay? So, in appearance, multinucleated, parallel, long cylindrical cells, and striations. The cardiac tissue it's kind of messy looking compared to the skeletal, isn't it? We call that branching. So right here, this is one cardiac cell that kind of goes off in two different directions. We call that, where it goes off in two different directions right here, we call a bifurcation. So this is the end of one cardiac cell right here and the beginning of another cardiac cell, and they're a lot shorter than skeletal muscle cells. Right here, where two cardiac cells come together, is what's called an intercalated disc. The very developed plasma membrane of the two cardiac cells forms that thickened area called an intercalated disc. And there's gap junctions. Remember when we talked about the membrane junctions, what holds two cells together to form a tissue? Well, when cardiac cells come together, they have gap junctions in there so ions can flow from one cardiac cell to the next cardiac cell very rapidly. And that's the signal for those cardiac cells to all contract together. Okay? And you can see striations. Okay? So we describe cardiac tissue as being the cells are bifurcated, intercalated discs are present, and striations are present. Uninucleus, one nucleus. You don't even see a lot of the nuclei in the cells because of the way the tissue is prepared, but one nucleus per cell when you see it in the cell. Sure. Oops, sorry. I'll get back to you, Nicole. Christina.
The disc, is that where the two, is that the portion where it looks like it's cut into a square, like two dots?
Yes, right here. And yes, the darkened pink line is the disc. Okay, Nicole?
It just looks like there's more than one nucleus.
Here,
Right at the bottom.
here.
At the bottom.
Oh, down
Yes.
here. Yeah, they are drawing more than one there, aren't they?
Yeah, one further.
Yeah, it's interesting that they've done that. They may know that there's actually two cells in that area, that this is the nucleus for that cell and this is the nucleus for that cell. But it is uninucleate. Yeah.
Yeah.
Interesting. And very astute of you to notice that.
Sometimes I surprise myself.
Okay, so the last slide we need to look at is smooth muscle, and you can see that there's some cylindrical cells in here, uninucleate, no striations. That's the biggest thing you should notice, or the most important thing you should notice with smooth muscle. There are no striations there. Okay. All right, questions about the different types of muscle tissue? You good? Okay.
Can I just ask one question? It might seem like a stupid question.
It will never be a stupid
When
question.
ions are transferring through the muscle,
Yeah,
is that diffusion?
it would be diffusion, yes.
Okay.
Yes.
I know, it sounds like a stupid question.
Yes, it is not a stupid question. It is not at all a stupid question. I am just going to, hello, hello. Maybe I can't do that. Really? Oh well. So that leads us back to where we were before I went someplace else. And here we have a list of functions of skeletal muscles. And you know how I like to ask you about functions. Skeletal muscles produce movement. They maintain posture, stabilize joints, and generate heat. We know it's pretty easy to figure out how they produce movement. We ask our muscles to contract, and I can lift my clicker. Maintain posture. Just sitting in a chair, like you're doing now, requires that your postural muscles be contracted to a certain degree. If you don't have an appreciation for what that takes, if you've ever had major surgery and you may have only been lying around for less than 24 hours because they like to get everybody up very quickly now. The first time they ask you to get up, you've got the poles and the lines and the Johnny, and They're just going to ask you to take a few steps and sit in that chair that's next to your bed, and you think, I'm not sure I can do this, okay? That tells you that your postural muscles require some energy. Your body's now paying attention to the surgery you had. It's trying to repair your body, and a lot of your energy is going there. You've lost a little bit of muscle tone in the short period of time that you were just laying in bed, and now you need to get those postural muscles to contract. They're very important in terms of getting you to sit in a chair, and even more difficult, stand up. That's why at the end of the day we're tired, especially if you have a job where you have to stand most of the day. You're asking your postural muscles to work all day long and not giving them a break. Stabilized joints. We've talked about the fact that tendons cross joints. They do that to keep muscles, not muscles, but your joints, from moving in directions that you don't want them to. You only want your knee to, and we'll talk about the words I'm going to use now, you want them to flex and extend. Flex and extend. You don't want your knee joint to twist, okay? And the tendons that you have crossing those joints, which are attached to muscles that are contracting a little bit, help to keep those muscles moving in only the directions you want them to. When we ask our muscles to contract, not all of that energy goes into doing work. Some of it, there's a byproduct called heat, and that's a good thing because we need to maintain a certain body temperature. So it's the contraction of our muscles that keep our body in the temperature that we want it to be. What do we do when we're cold? Shiver. That is quick contractions of your muscles to get some heat generated. All right, now we've talked about the gross anatomy of muscles. We've talked about what the muscle group looks like and the fact that, yeah, Penny?
The muscles that we're talking about right now are the muscles, right?
Oh, we're talking about all of them.
Okay, but the skeletal muscle is the first layer of muscles.
Actually, there's many layers of skeletal muscles. You've probably got 3 layers. You can take off the outer layer of skeletal muscles. Under there's another layer, and then there's another layer underneath. So, and then most of your smooth muscles are internal. They're in visceral organs. Okay, so I wouldn't call it another layer. They're just located someplace else.
Years ago, I woke up and I couldn't straighten my legs out.
Mm-hmm.
You know, I was like in law, and 2 weeks later, $3,000. My doctor said you need to get a photo of my skeletal muscles.
Wow.
And then, but for 2 weeks, they got me every test there was, and find out what was wrong with me. He gave me Appleton.
Mm.
And that's what he
Yikes.
said, because I was losing the nail, so I was always pulling my nail forward.
Oh, yeah.
So,
Yep. Repetitive motion. Mm-hmm. You pulled them, or they cramped.
to me, it felt like I was just like a cramp. I couldn't straighten
Right.
it out.
Yeah.
But after all the tests they took,
Yeah.
you know,
Huh.
incredible test, he just told me that they couldn't find anything that, you know, died.
Yeah.
You know,
Muscle
they thought
relaxant.
that It was that little thing that drops down right here that I could twist it back.
Ew.
It wasn't that.
No.
So finally, yeah, 2 weeks of not being able to straighten out, they gave me Appleton and said I pulled my skeletal muscles.
Hmm.
And
All of them?
about 2-3 days, and yeah, all
Wow.
2-3 days, and I could move around.
Oof. Yikes. In those instances, it's hard to get to the doctors.
Yeah, but then I thought about reaching back and pulling that toenail,
Mhm.
back and pulling the knee. That's why I wasn't doing just one side. I was doing
Both sides.
bringing that nail forward like
Mm.
that.
Wow. Yikes.
It hurt.
I bet. Okay, so we've talked about what we can see with the naked eye. Now we're going to have to use the microscope. And we've done a little bit about the microscope. That's when we looked at the tissues, we were using the microscope. Now we're going to magnify even further to see what's inside the cell. Before we do that, let's talk about some terminology. We have a special name for the plasma membrane of muscle cells. It's called the sarcolemma. So when you see the word sarcolemma, you're going to have to, in your mind, switch to, oh yeah, that's the plasma membrane. We just have a special name for the plasma membrane of muscle cells. Inside the muscle cell, we have these protein fibers that are called myofibrils. Long organelles inside the muscle. They're made of proteins. So we'll talk about the myofibrils. We're going to learn that there are 2 different myofilaments. And there's lots of M words here, and I will organize that for you so you know where we are. There's also a sarcoplasmic reticulum. It's specialized smooth endoplasmic reticulum. This is why we spent so much time in Chapter 3. So by the time we get here, you're not overwhelmed with needing to know what a smooth endoplasmic reticulum is. You know that it is a cellular organelle, and it's mostly involved in making lipids and carbohydrates. And we're going to call it something special in the muscle cell, a sarcoplasmic reticulum. Remember that slide a little while ago that said that when we're talking about muscle tissue, there's going to be some prefixes on words, MYS, MYO, and sarco. Here we're exposed to 2 sarco terms and a myo term, all right, all meaning something pertaining to muscle tissue. So now I want to, first of all, go back to a previous picture. I'm going to end this and show you what we're looking at. I'm going to end show, and we're going to go back to an earlier diagram because we are getting magnified. And I don't want you to, oops, I could have used this one. I don't want you to lose track of where we are. This is the whole muscle. There are some muscle cells in there, muscle fibers in there that are bundled together in fascicles, and inside the fascicle are individual muscle cells. See how we've pulled out one muscle cell at the top? Okay, everybody with me? Now I'm going to fast forward, and we're going to just look at what's inside a muscle cell. Okay, this one muscle cell, okay? This muscle cell is multinucleated, and if you look inside, it's full, bless you, of these protein fibers called myofibrils, okay? So what's sticking out here is a myofibril. This is not a muscle cell. This is the entire muscle cell. This is in the cytoplasmic area of the muscle cell. Muscle cells are specialized cells that have pushed a lot of their organelles to the side, and their entire cytoplasm is full of these contractile proteins called myofibrils. The way, see how these myofibrils have some straight lines here, red lines, and this red line going in that direction? Having all of those line up is what creates the striations in muscles. So we have what's called alternating dark and light bands. This dark band is this area right here where you've got lots of those special myofilaments, which we'll name in a minute. And then see this area right here that has a lot of white on either side of it? That's the light band. So we have dark light, dark light, dark light. That's what causes the striations. When you look at skeletal muscle or cardiac muscle under the microscope, that's what's causing the striations. And of course, we're not just going to call them light and dark bands. We have a letter associated with them. The dark bands are called A bands, and the light bands are called I bands. The way I remember this is that the letter A is in dark and the letter I is in light, and they don't have the other letter in them. So that's just a little mnemonic device for knowing that dark bands are A bands and light bands are I bands. Okay? So, and then it's also showing you the plasma membrane of this cell, and remember we call that the sarcolemma. All right. With me? All right. Now, the microscopic anatomy of skeletal muscles continues by telling us that the fibrils are aligned to give distinct bands, I bands or light bands, and now they're giving us some new information. The I bands only contain this component called a thin filament, and the A bands only contain thick filaments. So now we're going to take, now let me just go back so everybody knows what I've done. Now what we're doing. Now we're just taking a myofibril. This is the entire muscle cell. Now we're just going to look at one myofibril. And what the myofibril is made up of are thick filaments which are always going to be color-coded red in your book, and thin filaments, which are blue. The thin filaments, the blue ones, are made up of the protein that's called actin. The thick filaments are made up of a protein called myosin. Collectively, we refer to the actin and myosin proteins as myofilaments. And so at this stage, usually most of my students are going, okay, there's too many M words and I'm not sure where I am anymore, okay? So I'm going to change to a white screen, and I'm going to, in a flow diagram, I'm going to change to a felt pen and a white screen, and let's organize everything. We start out with a whole muscle. That whole muscle is organized into a fascicle, isn't it? Which is bundles of, that's an E, bundles of muscle fibers. If we look inside those fascicles, we're going to find muscle fibers. SC, muscle fibers. Oh gosh, let me see. Try that again. And remember, a muscle fiber is really a muscle cell, right? Inside the muscle fibers, we have myofilaments. Myofibrils. Sorry, I got ahead of myself. Myofibrils are made up of two types of myofilaments. They are actin and myosin. Myosin. And actin is also called the thin filament. And myosin, ignore that line, myosin is called the thick filament. I wonder what happens if I go back and just want to redo that, get that out of there. Oh, good. How about that? Hey. And then actin has an N on the end of it. So there you go. So we've actually made quite a lot of headway into understanding the anatomy of muscles. We started with the picture of the whole muscle. Muscle cells are organized into fascicles. You go inside the fascicle, you find a muscle fiber, which is a muscle cell. If you look into the cytoplasm, and maybe I should write that next to myofibrils, maybe it will help you to know where we are if I put in cytoplasm of the fiber. And then we saw from the diagram of the myofibril that it's made up of actin and myosin. Actin and myosin are referred to as myofilaments. Okay? Actin is always called the thin filament. Myosin is called the thick filament. Everybody with me? Okay. We still have that. Okay, so let's go back and look at this again. The myofibril has both thin filaments, which are always colored blue as we go forward, and thick filaments that are red. Thin filaments are actin, thick filaments are myosin. Okay.
Is that going to be an exam like that? Are you going to do the red blue thing?
I don't have any pictures of that. You're not expected to label it, but you are expected to know thick and thin and then actin, myosin, that designation. So one contractile unit of muscle is called a sarcomere. The contractile unit of a muscle fiber is a sarcomere. Organization of the sarcomere has myofilaments, thick and thin filaments. The thick filaments are made out of myosin, which is a protein, has associated with an ATPase enzyme. That will be important when we talk about how muscles contract. When we actually get into the muscle physiology part, right now we're still paying attention to anatomy. Myosin filaments have heads, extensions, or cross bridges, and we'll see those as we get more magnified. Myosin and actin overlap somewhat, and the thin filaments equal actin filaments composed of the protein actin, and they're anchored to the Z discs. So this is the same structure that we looked at in the previous diagram, but we're magnifying it more. So you can see the details of what actin and myosin look like. And we can also see that if you go from one Z disc to the next Z disc in the myofibril, that you have a sarcomere, which is what, as we go forward and explain how muscles contract, that's one contraction unit. So once again, I'm going to back up just so that you can keep track of where we are. Okay, this diagram has a Z disc here, and a Z disc, by the way, is where thin filaments attach to one another. The actin protein attaches to one another on this area called E disc, and then again here in a Z disc. And if you go from one Z disc to the next Z disc, you have one contractile unit called a sarcomere. You've got thousands of them. In the entire length of your muscle cell, you probably have thousands, if not 10,000 of these sarcomeres. We're going to explain what happens during contraction in that one area of a sarcomere. The same thing's going to be happening in all of the sarcomeres. We're just going to simplify things and talk about what happens in that one sarcomere.
A Z disc is made up of the thin filaments.
It is. It's actually where the thin filaments join to one another.
On our PowerPoint outline, it says it's anchored to the Z disc like it's a separate entity.
It creates the Z disc. Where the thin filaments connect to one another, it creates that Z disc.
That one's created
H. The H zone, I know there's some red lines going through here. Those are a protein, think of a sewing needle that attaches all the thick filaments together. The H zone, however, is defined as where, when the muscle's relaxed, there's no thin filament. That's the important thing to know about the H zone. And that's an important thing to know as we go forward. These red lines that are going kind of around are sewing up the myosin filaments. That's not what I want you to pay attention to in the H zone. What's important to know about the H zone is that when the muscle is relaxed, there's no thin filament. Okay? H zone is empty of thin filaments when the muscle is relaxed.
They connect when it flexes.
They get pushed in when it contracts.
So in other words, at rest, the avian, there is a zone that lacked actin filament. That's called the H zone.
Correct. That is correct. Alrighty then. So now we're back to our diagram of one sarcomere that has been magnified even further. And you have the blue actin filaments, which look like each actin protein actually is one of these blue spheres, and they're strung together like a string of pearls, and there's two of the string of pearls, and they're twisted on one another. That's what forms the thin filament. The thick filament is this red portion here, and it's showing you that it has myosin heads. The protein, the way the protein has folded, it's formed these myosin heads. What happens during muscle contraction is that there is a binding site on actin that the myosin heads are really attracted to. When the muscle's relaxed, the binding sites that the myosin heads want to bind to, they're turned, this actin molecule can rotate. When the muscle's relaxed, and it's not being asked to contract, the binding sites are oriented away from the myosin heads. When a nervous impulse comes down, a series of events takes place between the nervous system, the axons of the neurons, and the muscle. It results in the actin molecule rotating so that the myosin heads can now bind to the actin, and the myosin heads, once they bind, they push, and that happens on this side and it happens on this side. The myosin heads bind here and slide the actin filaments into that empty H zone. If you now take the thin filaments and push them in here and here, what happens to the length of the muscle? It shortens. So we're going to see more slides in more detail about how muscles contract, but in a nutshell, that's how they shorten. Actin rotates so that a binding site for the myosin heads can engage. The myosin heads ratchet the thin filaments on both sides into the H zone and shorten the sarcomere. If this is happening in all of the sarcomeres in the muscle cell, the entire muscle cell shortens. If it happens in many, many, many muscle cells in a muscle group, the muscle shortens. So when I ask my biceps muscle to shorten to lift my forearm, this binding and sliding is taking place in thousands and thousands of sarcomeres inside thousands of muscle cells to get that to happen. It actually turns out that it takes more muscle cells to contract, to lift, let's say, this table. I don't need to call up as many to lift just the clicker. So muscle responses can be, and contractions can be, what we call graded. Okay, we don't need all of them to participate. All right, so as we go forward, we're going to see more and more diagrams of myosin heads, actin filaments, and how the myosin heads actually get to engage into the actin. All right, microscopic anatomy of skeletal muscles. At rest, within the A band, there's a zone that lacks actin filaments. We call that the H zone, or the bare zone. We also have sarcoplasmic reticulum that in the role in the muscle, it stores a tremendous amount of calcium for us and surrounds the myofibrils. So here we have the picture again. They've just made it bigger. Okay, we keep getting bigger and bigger. So you can see myosin heads and the actin filaments. The bare zone, which is also the H zone. Okay, don't forget that letter designation. And when the muscle is relaxed, the actin is not in that H zone. It's at its full length. Before we continue our discussion of how muscles contract, we need to put some words on what we're going to talk about. So, in order to talk about the stimulation and contraction of a single skeletal muscle cell, we need to know that it has the characteristic of being excitable, meaning the nervous system gets to control what happens in the muscles. The nervous system piece of this discussion is going to be a big black box for the most part. That's the next chapter after the next exam, okay? I want you to understand what happens in the muscle. I'll give you a little bit of information as to what the nervous system does, but really don't feel too badly about the fact that you don't understand the whole nervous system piece, because we haven't done that yet. So the nervous system can actually excite muscles, which means that the muscle can respond to what the nervous system is asking it to do. We also say that muscle cells can be irritated. They have the characteristic of irritability because the nervous system can irritate them or excite them. That's all saying the same thing. Once the nervous system excites a muscle, it can contract. So we say that muscle cells have contractility, the ability to shorten. Contractility, ability to shorten when an adequate stimulus from the nervous system comes in and it's received by the muscle. Extensibility, muscles can also be stretched. Think about climbing stairs. Think about the gastrocnemius muscle in the back of your leg. When you're climbing stairs and that muscle's doing work for you, is it shortening? Pretend you're going upstairs.
Shortening and lengthening.
Pretty much it's lengthening. You're right. Actually, when you lift your leg up, but when it's doing the work, it's lengthening. So there is a way for muscles to do work, and there's tension on them, and it will allow you to climb stairs, but the muscle is actually lengthening. So that's what the extensibility is referring to. And then elasticity. Once we contract and shorten our muscles, we then want it to go back to its uncontracted state, don't we? If you think back to when we had sarcomere pictures up there, we had we had yellow parts at the end of the sarcomere. There were these yellow, they looked like springs. Those are elastic fibers. So when the nervous signal to contract stops, the elastic fibers spring all the sarcomere back to the default setting of not having those actin filaments in the H zone. Pulls the sarcomere back to its starting default position. Okay, that's what elasticity means. Lynn?
I had a broken hip like a week after, I had been in bed rest, like they had me doing exercises. My muscles started, I don't know what they told me they were spasms.
Yeah.
What really brought that on?
Spasms are going to be uncontrolled muscle contractions. In other words, it's not that your nervous system is asking them to do that. They're kind of doing it on their own. And they're probably doing it because they're out of position and they're irritated, okay, kind of a scenario. And it's just a spasm is basically the same thing as a muscle cramp, okay? And so the physiology is not right. That's probably been damaged to a certain extent. And they're trying to repair themselves and work around the fact that they're not in the right position, They're really kind of confused. I bet.
Until they were able to give me muscle.
Yeah. They didn't want
I
to.
was using the muscle.
No.
It was just brought on.
And sometimes not using the muscles will bring that on too. Sometimes having them in the wrong position or injured in your case because you had a fracture. So the bones were probably not lined up correctly. And they're trying to get that to happen. The muscles are trying to get your hip to be realigned appropriately. And to do that, they'll spasm. All right. Any other questions about the definitions that you see relating to muscle contraction? All right, so the nerve stimulus action potential. Here's where we're going to briefly see that nerve cells, the processes from nerve cells end on muscles. Skeletal muscles must be stimulated by a motor neuron or a nerve cell. A motor unit is one motor neuron in all of the skeletal muscle cells stimulated by that neuron. So this piece of the nervous system is kind of a big black box, but this is what your nerves look like when they end at a muscle cell. They end in these bulbs called the axon terminal, okay, and this is where the chemical called a neurotransmitter resides. It's the release of neurotransmitters that actually get the muscles to contract. They actually set into motion the events that need to take place in order to get the myosin heads to bind to the actin. So I want you to understand that motor axons, nerve cells, end on muscles, and when the nervous system fires, it can get muscle cells to contract. Okay, and we'll look a little more, and this is actually an actual slide, an actual micrograph of the nerve endings on the muscle. Okay. Nerve stimulus and action potential at the neuromuscular junction. Let me go back one and explain to you where the neuromuscular junction is. Where the end of this nerve cell ends, see all the axon terminal ends that have the neurotransmitter? Where that is up against the muscle fiber, that is your neuromuscular junction. Nerve cell ends with all these little bulbous ends, muscle fiber is right under it. There's a small space called the synaptic cleft, but that is your neuromuscular junction. So let's, let me turn this up. Let's go see if we have time to watch this. I need to change to an arrow. And by the way, you have this available to you up on the content area of Mastering A&P. I think it's still loading. These little flicks take a while. I think we'll just have time to watch that, and then we'll be done. If I can get it to load. It's still flashing, so I still think it's loading. There we go. I think we're close. We're close. Okay, still thinking. Unbelievable. Let me try again. Oh, here we go. Great. Now I'm going to have two of them. All right, hold on. We should be close. Of course, there's no volume because I will start it over again. For some reason, my volume's down at the bottom here.
Neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber.
All right. Turn that down just a little bit. All right, here we go.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, that's where this story ends. The part we haven't seen yet is when that action potential, or the wave went across the sarcolemma. It starts some more events that lead to the myosin binding to the actin. I want you to read the rest of this chapter because I don't want you to be overwhelmed as we go forward, okay? I think you might be overwhelmed at this point. Get into your book. The homework is already up on Mastering. Work on that, and we'll finish the chapter on Wednesday, okay? See you then.